Okay, so here is the last set of questions from Chapter 15. Again, the specifics about Chapter 15 I talked about in the first video. Just to kind of reiterate, transcription and translation. There's some multiple choice, one matching question that there's only a couple of answers to. And then, again, for the short answer, really, you want to know RNA's structure and function, but there's no specific question there on the steps in transcription or translation. There are in the multiple choice. You want to make sure you know RNA polymerase is involved in transcription. You want to know about maybe RNA processing. You want to know about the different types of RNA. Okay, so let's go ahead and jump into these. All right, so the first question, and I'll tell you that I don't provide you with this table. There's no questions about the genetic. There's no specific questions on the genetic code. So don't worry about, I've said this before, but don't worry about memorizing this table. Don't worry about memorizing amino acids. Don't worry about memorizing codons or anticodons. You should know what a codon is and what an anticodon is, but you don't need to memorize the specific ones. Okay? So here's a question. It says both AUC and AUA are codons in mRNA that both specify the same amino acid, isoleucine. And so what feature of the genetic code explains this phenomenon where you have two codons that code for the same amino acid? And so C, the genetic code, D, the genetic code is redundant. Okay. C is also true, it's universal, but that's not what this question is specifically pointing to. This question is pointing to the fact that it is redundant. You have two codons specifying for the same amino acid. So that's redundancy. Another word sometimes used for redundant is degenerate, and that means the same thing. Okay, I don't love that word, but sometimes we use that. Okay. Next question says, how many nucleotides are in 12 mRNA codons? So you have to know how many bases is a codon, and then you gotta do the math. So I'll let you think about that for a minute. So hopefully you said C, 36. So there's 3 nucleotides in a single codon, so 3 times 12 is 36. So there will be 36 nucleotides in 12 mRNA codons. So again, it's a triplet code. The mRNA is read 3 bases at a time. 3 bases make up a codon, and that's how the ribosome reads it. Okay, so here's using this table, just making sure we understand this. This is something that you'll have to do in lab. If 6 bases within a gene are these, what will the mRNA transcript read, and what will be the amino acid sequence? So I gotta write this down. Give a minute to look this up. Okay, so it looks like the message in B and in, no, just B, right? Yeah, the only one that's correct is B. So your mRNA should read 5 to 3 GUA, GCA, and let's make sure that those are the right 2 amino acids. So the first codon would be the first 3 bases, so GUA, so GUA codes for valine. Okay, and the second one is GCA. So G, second base is C, and A. So alanine. So the correct answer here would be B. Okay, so the next question, Okay, it says knowing that there are 20 different amino acids, and yet there are 64 codons. How many potential proteins are there? And so, yeah, the math here, you can't do it because genes are different lengths, right? So mRNA molecules are going to be different lengths. It doesn't matter how many different amino acids there are or how many different codons there are. There's an infinite number of proteins that can be made, okay, depending on the message. So C would be the correct answer. Okay, the next question says, in transcription, so going from DNA to RNA, the promoter region. What's true about the promoter region? So A would be the only one that's correct. It contains the consensus sequences. And actually, that you can see that in this picture here. You can see at the -10, so before the gene, before the transcription start site, you have what's called the TATA box. And then at the -35 region, you have another consensus sequence. Those are found upstream of the gene, not downstream. So before, not after. So B is not correct. It's not involved in termination. It's involved in initiation. And D, where the ribosome binds. So when we talk about the ribosome, we're talking about translation. This is transcription. And so that's not true. This is where RNA polymerase binds, not the ribosome. So that's different. So A would be the only correct answer there. Okay, transcription ends. I'll give you a second. And so the correct answer there would be C. Okay, so no, it's not upstream of the promoter sequence. It's downstream. It's at the end of the gene. B is not correct. Elongation is when the RNA transcript is growing. So that's not termination. And D, it says once the RNA polymerase reaches the end of the chromosome, not necessarily, maybe, but it's when RNA polymerase reaches the end of the gene, which can be anywhere in the chromosome, not necessarily at the end. And so C is the only one that is correct. RNA polymerase will reach a region at the end of the gene where there's some type of a termination signal. Okay, another question on transcription. So it says, which feature of transcription promoters can be found in both prokaryotes and eukaryotes? And so again, I'll give you a second on that one. And so the correct answer there is B, the TATA box. That's a common consensus sequence in all organisms, which is used during initiation for that RNA polymerase to bind. The -35 sequence is not present in eukaryotes. Start codon, has nothing to do with transcription, and it has to do with translation. And the GC box is not something. Here we go. So eukaryotic and prokaryotic transcription differ in that eukaryotic transcription so C would be the correct answer. It occurs in the nucleus, and it involves more enzymes, more proteins, more transcription factors, it's more complex. The rest of these answers are not correct. So A is not correct because it says that transcription can occur while DNA replication is proceeding. That doesn't happen. B says this occurs simultaneously with translation. That can't happen in eukaryotes. That's true for prokaryotes, but not eukaryotes. And then D, it doesn't necessarily take longer. Sometimes it will. But the second part of this is really wrong. It results in more mistakes. Not necessarily. RNA polymerase is pretty efficient. Okay, so C would be the only one that makes real sense. Ooh, wrong way. Okay, this is about RNA processing. So it says during eukaryotic mRNA processing, remember, mRNA processing only occurs in eukaryotes, not in prokaryotes. So in prokaryotes, RNA is made and it's read. There's nothing that happens in between. In eukaryotes, you have a pre-mRNA that then gets processed, and you end up with an mRNA that will get exported from the nucleus and read by a ribosome in the cytoplasm. Okay, so let's see which one is true. Okay, so it would be B. So why is A wrong? So A is, if it said introns are removed and exons are spliced together, that would be correct, but it reversed those words, so that's not right. Exons are the parts that we keep. Introns are the parts that we cut out. 5 is wrong because the poly A tail is added to the 3' end, not the 5' end. And D is wrong because, yeah, the mRNA eventually gets exported to the cytoplasm, but that's not part of RNA processing. So the only one that is correct is B. At the 5' end, we add the cap, which is made up of guanosine. Okay. Okay. The 5' cap. So which one is true here? Give you a minute. So B. Once it gets out to the cytoplasm, the mRNA, there's lots of enzymes in the cytoplasm that can degrade mRNA. Your cells actually have a natural defense mechanism to get rid of mRNA, especially non-eukaryotic self mRNA. So there are enzymes in the cytoplasm that can degrade this. And so by having both the 5' cap and the poly A tail, that's going to prevent degradation or the breakdown of the mRNA. It's going to help it to last longer in the cytoplasm so that proteins can be made. And it also assists in translation. Okay, so B would be correct. It helps to promote ribosome binding. So B. Okay, this is the process by which amino acids are joined together according to the sequence of bases in mRNA. What does that describe? That's translation in a nutshell. Okay, the ribosome translates the message, and as it does that, it's going to bring in tRNA molecules with the right anticodon. Those will hold the appropriate amino acid, and we end up building a polypeptide chain, which eventually folds up into a protein. So that is translation. Okay, so we talked about the codon being a triplet of bases in the RNA, and this picture is beautiful. You can see right here in orange, this is the RNA, and you can see 5' to 3', and it's being read in that direction. So the first is AGG, and you could see the tRNA molecule in here. If you look at the base of it, the bottom of that tRNA, you see the anticodon, ACC. Next triplet, UC, that's the codon, and the anticodon is here at the base of the tRNA, AAG. It's complementary. So the anticodon and the codon are complementary to each other. So this says the anticodon. What's true about it? All of these. So yeah, it's found in the tRNA at the base of it, right? It is going to be complementary to the codon in mRNA. And third, it specifies the amino acid that will bind to the tRNA, right? So if I come out here and I look at these molecules, these tRNAs before they bind to the ribosome, the little blue circle in this case, or diamond, whatever is attached to it, that's the specific amino acid. And the amino acid that binds is specific to the anticodon, which is complementary to the codon. And so that's how we make sure the right sequence of amino acids ends up being built. It's built on the message that originated in the DNA, that ended up in the codons of the RNA, and the anticodon of the tRNA ensures that the right amino acid is brought in. So here we go. A scientist introduces a mutation that makes the large ribosomal subunit non-functional. So the large ribosomal subunit can't bind. It doesn't work. What will be the effect on translation? So take a minute to think about this one. So you have to know what happens during initiation. And so the correct answer here would be A. Okay. B is not correct because the process isn't even going to get started. Elongation doesn't get started because initiation doesn't finish. So B can't be correct. C, it says the ribosome won't be able to interact with the mRNA at all. And so that's not true because the small ribosomal subunit actually binds first. And it doesn't need the large ribosomal subunit to bind. And so that's going to happen. And then D says tRNAs will not be able to exit the E site. Well, they won't be able to bind at all because, or they won't be able to bind to the E or the A or the P site because they won't exist because the large ribosomal subunit won't be there. But what will be able to happen is the small ribosomal subunit will be able to bind. It'll be able to find its way to that initiation codon. The first tRNA molecule will probably come in, right, with its methionine bound to it, that first amino acid. But then that's it, because the rest of the process requires the large ribosomal subunit to be present. And so that would be A. A is the correct answer. Okay, I like this question too. It says many antibiotics inhibit bacterial protein synthesis, which that's true. For example, if you've ever taken the antibiotic tetracycline, tetracycline is an antibiotic that's oftentimes used to treat acne. Tetracycline blocks the A site, the aminoacyl site, on the bacterial ribosome specifically, which is neat because it doesn't affect our ribosomes. It only affects the bacterial ribosome, so we can really target bacteria. There's a second antibiotic called chloramphenicol, and that blocks peptidyl transfer. Okay. What effect would you expect each of these antibiotics to have on protein synthesis? So the first question's on tetracycline, so think about just that one. So tetracycline would directly affect what? So A would be correct, right? Because if the A site doesn't work, if it's blocked, then tRNA molecules can't come in. That's where they come in to the ribosome. So that would be A. And the follow-up question, this is the same question again, except for now we're looking at chloramphenicol. What would that directly affect? So I got to look back. That blocks peptidyl transfer. So which one of those would be affected? So that would be C, growth of the polypeptide chain. If you can't transfer the polypeptide chain, it can't grow, right? It has to be transferred during this process to grow. That's it. Okay. So again, I'm sorry that we couldn't do this live. I wanted this to be equitable for everyone, and so I figured recording myself was the best choice here. If you have questions, I have, office hours tomorrow, which is Wednesday, office hours Thursday. And for those of you who are in my Tuesday or Wednesday class, I also have office hours next week. So please feel free to reach out with questions by email or ask in class or during office hours. Good luck making your cheat sheet. Hopefully I've narrowed things down a bit for you.